Good morning. I want also to thank the organizers for inviting me to this very exciting meeting. I'm really looking forward to the next 2 days when we are going to discuss some of the very critical ethical issues that are coming up now with advances in research. I'm going to tell you about some of the work that we've been doing on germ cells. So these are cells that gives rise to sperm and eggs, and the particular investigation that we are doing is to see if we can make synthetic gametes from somatic cells, which don't normally make germ cells. And this raises ethical issues, but it also opens up many opportunities, and that's what I'm going to tell you about. So I want to start by telling you something about one of the pioneers of germ cell research, which was Weissman. And this is important because what he told us was that there is a fundamental decision that's made very early in development, and that is to do with a cell either become a somatic cell or a germ cell. This is quite a fundamental decision that's made very early in development. And what Weissman actually put forward this idea, and it was quite important at that time, is that the cells that give rise to the body, the rest of the body, will perish with each generation. So you have a cell which will divide and either become germ cell or a soma. And these cells will undergo aging and they will die. Whereas cells that give rise to germ cells and become either sperm or eggs will generate a totipotent state. And these are the cells that will give rise to the whole organism. And this lineage is considered to be totipotent because it gives rise to endless generations. So any information that's passed down this lineage continues through generation after generation. So that's the important point to remember, because any change you make to the germ line will continue and will be transmitted to subsequent generations. So why is it important that we study the germ cell lineage? So there are several things that are important here. So the first thing is that the germ cell lineage is the only one that can create this totipotent state. It's the lineage that can give rise to the whole new organism. And this occurs through formation of these cells. They're called primordial germ cells, which are formed very early in development, which give rise to sperm and eggs. And when you get fertilization, you get a new organism. And the germ cell lineage is considered to be immortal because it's the only lineage which gives rise to one generation after another, whereas the rest of the cells will die out. Now, the other important point about this lineage is that it's the lineage which transmits information to next generation and to subsequent generation. And the last point, which is extremely important, is that it's the source of genomic innovation and evolution. And I'll come to that towards the end. It connects with what Arnold was talking about. Okay, so I want to tell you very quickly about the origin of where do these germs originate. And they originate very early in development. So here's a mouse embryo which is developing from one cell, pre-implantation embryo implants. And this is quite an early post-implantation embryo. What you find is that you get a cluster of about 30 cells. These are the founder cells which will give rise to the sperm and eggs when the embryo becomes adult. So this is a cluster of about 35 cells, which are very important. And these cells are set down and are going to eventually transmit the information to all the subsequent generations. Now, some years ago, we wanted to know how these cells become specified during development. So we did a number of experiments. I won't go into the details of it. But at the end of these experiments, what we found was that you need three key genes, which are shown here, PLIMP1, PID14, and AP2-gamma. So we have about 20,000 genes, and you only need three genes. These are three key genes that you need to make this founder population of primordial germ cells. And we know that these are key genes because if you introduce a mutation in any of these genes, you will abrogate formation of these primordial germ cells. So we've come to understand a little bit about how these very important cells are laid down at the very start of development, which will give rise to sperm and eggs. Now this knowledge, this has accumulated over the last 20 years or so, has also led to new developments, which is the ability to make germ cells from embryonic stem cells. So this is, again, work in mice. So we can take this blastocyst, make these pluripotent stem cells, and by using a variety of signaling molecules, we can prepare them so that they become responsive to particular signals to form these primordial germ cells. So these are now pluripotent stem cells, and we can take them in culture, grow them, and by using a number of signaling molecules, can induce formation of germ cells. And we can make endless germ cells in culture using this particular technique. So this already raises some quite interesting questions. So it's not only that we can make these germ cells, but I think the key point that I want to go back to, which is the Weissman's idea that you have two types of cells, a soma and a germ cell, but we can now actually break this Weissman's barrier. Because what we can do is to take these somatic cells, we can reprogram them to make them into this induced pluripotent stem cells, or IPS. And using this IPS, we can make primordial germ cells and then generate this totipotent state. So this is quite a profound change. If you think about it, what this actually means that potentially you can take your skin cell, you can reprogram the skin cell, make them into primordial germ cells, and you can theoretically make a whole organism from that. So this is conceptually and ethically is a quite an interesting idea, and I think it's something that requires a lot of discussion. Now, it's not only that you can make very early primordial germ cells, but a group in Japan actually uses very similar technique, starting with pluripotent stem cells and also IPS cells, and made these primordial germ cells, and then eventually they made also sperm and eggs. And from these sperm and eggs, you can make synthetic embryos and give rise to viable young. So you have now taken a skin cell, turned it into sperm or eggs, fertilized them, and made a whole organism. So in the last 20 years, from the very beginning of just understanding how these cells get specified, we've reached a point that we can now make viable young from these somatic cells. So that's the work that's been accomplished in the mouse. So what about the human? So you're now trying to come to understand how the human germ cells are formed. There's been a long quest to try and understand the origin of human germ cells, and there was some work that was done almost 100 years ago, where people were trying to find out where these germ cells originate. And they were able to find that the human germ cells in an embryo that was 3 weeks old. But it's only recently that we have started to really understand how the human germ cell lineage starts to form. It's only in 2015 that we started to find out about the human germ cell lineage. Now, it's important that we study the human germ cell lineage because there are differences between mouse and human. In the early development, in the way the pluripotency network is set up, there are differences between mouse and human. And also, if you look at the early embryos, the mouse embryos develop as egg cylinders, whereas the human and many other mammalian embryos develop as bilaminar discs. So the organization of the embryo is not similar. And this has an effect on how cell fate decisions are made, and on the mechanisms of cell fate decisions. So now we think that the human germ cells probably arrives at around day 17 of development. The problem is that there are current regulations which prohibit investigations of human embryos past the 14-day stage. So it means that we can't actually grow these human embryos long enough to see where the germ cells originate. So we have to actually think about other ways of looking at this, and we have to now turn to using human embryonic stem cells or induced pluripotent stem cells, which is what we did, putting in a reporter and try to make human germ cell-like cells in culture as we did with the mouse system. Now, the important thing is that we have to have a reference point, and we have to take the real germ cells from human fetuses to know whether what we are making in culture resembles something that's occurring in the embryo itself. We can get these fetal embryos from around 4th week of development. These are quite early, but usually we get them at around 7 weeks. So we made these germ cell-like cells, and then we compared them with fetal germ cells. Now, I won't go into the detail, but we can take these pluripotent stem cells and we can drive them towards development which resembles what happens in the early embryo. And at some point in the middle of this development of these cells, they reach a point where they become responsive to signals, and they will start to make this human primordial germ cells. We can see that by looking at this reporter. So we can now make these very early human primordial germ cells. And when we look at the genes that are required for making human primordial germ cells, they're not the same. There are differences between mouse and human. I won't go into the details of it, but there are clear differences between mouse and human. But we can do very similar experiments to what we have done in mice. We can take this some of these key genes, and if we express them in certain cells, we can make endless primordial germ cells. So we can make large numbers of these very early human primordial germ cells now in culture. So this is the situation currently. So we can take these skin cells, we can reprogram them, or we can take ES cells, and we can induce these primordial germ cell-like cells. And we can make thousands and thousands of these, limitless. So it allows us to do quite a lot of experiments to understand the key properties of these cells. Of course, we would like to take them further. The immediate objective we have is to take them up to about the 9th week of development for various reasons. And that's something that's in progress. So we are trying to push them to grow further, and we're making a little bit of progress. There was a recent report from Japan showing that you can take these cells and culture them, and they will make oocyte-like structures. So we made quite a lot of progress with these early human germ cells. We are also trying this organoids, this Arnold mentioned. So what we're doing is to making these primordial germ cells, and at the same time, we are taking human ES cells and we're making these gonadal cells. We mixed them together to make this gonadal organoids, or we call them germinoids, to see if in this kind of germinoids we can get these primordial germ cells to develop further. And again, we have managed to get further development, but this work is at an early stage and it's ongoing in the lab. Now, I want to just touch on some of the properties of early human germ cells and why these studies are important for ethical reasons, and also because they tell us something quite interesting about humans and human evolution. So we can take, as I mentioned, we can take these early human embryos, which we can get around 4th week onwards up to about 22 weeks. And we can retrieve these from germ cells, developing germ cells from these human fetuses. And we want to look at the properties of these cells, how they are actually developing. This information is very important because we start to make artificial germ cells or gametes. We need some reference points. So we need to know how the germ cells actually are developing in the embryo itself. So when we make them, we can see whether what we are making resembles something that's happening in vivo. What we've been looking at particularly in these ex vivo germ cells is to look at changes in their gene expression, which is quite important because they are not static, they're always changing, so we want to know what kind of genes are being expressed. And the other thing that we are particularly interested in is what is called epigenetic. So these are instructions that are put on top of DNA, which instruct the cells which genes are going to be expressed and which are going to be repressed at a particular time in development. So it's very important that we understand what this epigenetic information is. But what happens in the germline is the resetting of this epigenome is very important because I told you that this lineage is in a sense immortal and is going to give rise to a new organism at fertilization. And there are some very, very important and critical epigenetic changes that take place in this very early germ cells in order to ensure that at the end you will make a totipotent zygote and a whole organism. So we have, I'll just give you one example of the kind of experiments that are ongoing. So we've been looking at, particularly at DNA methylation. This is a very key epigenetic mark, and quite uniquely in the germline, this mark starts to get erased. It's completely erased, and this doesn't happen anywhere else in any of the other tissues. And there are various reasons for this, but this erasure process is extremely important, and we've been looking at this. And just to give you a very quick summary of it, if you start with these pluripotent stem cells, or you look at the germ cells, what happens is that these bar plots tell you that these cells are starting to lose their DNA methylation. These are germ cells at different stages of development. And when they reach around the 7th or the 9th week of development, all of the DNA methylation is virtually wiped out. There's almost complete erasure of this, so which you call epigenetic ground state. And the germ cells have to reach this epigenetic ground state, which is prerequisite for generating totipotent state at the end. Now, while bulk of this DNA methylation is erased, there are some resistant loci that actually maintain. Although this is an acutely hypomethylated state, there are some regions that retain methylation, which is quite intriguing. But I think it leads to another very interesting point, what I want to end with. So as I told you, there are these resistant loci. And these resistant loci are made up of young transposable elements. I'll tell you what transposable elements are in a second. And what we find at these transposable elements, resistant transposable elements that we pick up in the human germline are actually conserved mainly in apes, but not in other mammals. So in other words, there are these resistant loci which have been transmitted through the primate lineage for around 20 or 30 million years. Now, what is also important is that the bulk of what is being transmitted by the germline is mainly what is called junk DNA sometimes, or we can call it the dark matter of the genome. It's only about 2% of your genome that actually codes for actual proteins. The rest of it is non-coding DNA. So what you're transmitting to your progeny is the bulk of the DNA is not coding, it's not making proteins, it's this non-coding DNA, what we call dark matter. It's mainly made up of repetitive elements and transposable elements. So what's being transmitted are these transposable elements that are resistant to reprogramming. Now, these transposable elements have been quite important and have played a very crucial role in genome innovation and evolution. And that's why these resistant loci are particularly interesting, because they might tell us something about human evolution, and that's what we are particularly excited about. So I'll give you one example of a transposable element. This is a transposable element called LTR12C. This non-coding transposable element entered into the primate lineage about 30 million years ago. It's not present in old world monkeys like macaques. It's only present in great apes. So this particular transposable element has been transmitted over 20 or 30 million years, being for a very long time. And what the hypothesis, and there is some evidence for it, is that during this course of this transmission of these kinds of elements, they go through mutations and they acquire functions. And this particular element, LTR12C, there is some evidence that this LTR12C might have acquired a particular function, because we now look at the human fetal brain, and what we find is that there are about 1,200 of these located in the human fetal brain. And they contain this histone modification mark, which is H3 equated H7. You don't need to remember it, but what actually this mark implies is that it has a potential to act as a regulatory element. So what has happened during evolution is that there are these kinds of elements that have got inserted into the brain, in this case, and other tissues, and they've acquired a new function. So we can see this if we compare, for example, human particular chromosomal region in the human chromosome and compare it with the macaque. What we see is that there is this introduction of this LTR12C, which has now acquired a function as a regulatory element. And what this insertion would do is to change the way the whole regulatory element, the whole of the transcriptional network will work. And this leads to the innovation of how through this kind of insertions, you get new functions can arise. So this comes to the question of what makes us human. And Arnold talked about the changes that have taken place in the developing brain amongst different mammals. Now, we know that if you compare a chimp with a human, we are very similar. We have 97% identity in terms of our genetic makeup. So there's a very small difference, but if you look at the brain of the chimp or the face, for example, it's quite different. And we believe that some of these non-coding sequences which have got evolved and got inserted have made a contribution to evolution and the complexity of the brain. And also, the other remarkable thing about humans is the face. And so these kinds of events have taken place. These transmission of this non-coding DNA over millions of years have led to this evolution and innovation, which has led to how the human brain has developed. So I think the germline has got quite a great deal to tell us, and that's why this research is very interesting. So I told you about how we can start with the zygote, which is a totipotent give rise to primordial germ cells, which will, once they start developing, they migrate into the developing gonads and start to become sperm and eggs. And then they will transmit information to the next generation. So this cycle just goes on, and that's why this is called an immortal lineage. But importantly, the germ cell lineage also transmits this non-coding DNA. The large amounts of DNA that is transmitted is non-coding. 98% of it is non-coding. But this has led to evolution and innovation. Now, the very last thing I want to leave you with is this is kind of the broad perspective of how we think this kind of research can be done in the future and how it can be used. So the idea is that we would like to eventually reconstitute the entire human germ cell lineage in culture, starting with stem cells or iPS cells, making first the primordial germ cells, getting them through this very important epigenetic programming event, making germinoids, and through these making gametes, viable gametes. We are only starting to scratch the surface at the moment, but we can see some signs that we are on the way towards this. So this will raise quite a lot of ethical issues. It's possible, potentially in the long term, we might be able to make gametes, which may originate from skin cells, from reprogrammed skin cells, make a viable zygote with potential to create a new organism. And we can make new ES cells and we can continue with this cycle, or we can use these cells to make somatic cells. Now, the importance of making these kind of model in culture, because we can't really do these kinds of experiments very easily in humans. So what we'd like to do is, if we can reconstitute this kind of a cycle, we can make synthetic gametes, so we can see how well they resemble the real gametes. We can also test gene editing. There are new tools that are coming up that we can try gene editing in this kind of manner and make somatic tissues, brain or liver or whatever we want, and ask whether this gene editing techniques we are using, how good they are. Do they make mistakes? Are they good enough? Can they be used in any way to correct human diseases? I haven't talked about epigenetic inheritance, which is a hugely interesting topic. This is whether the environmental factors can induce changes in your germline, what you eat, and all of those things, whether some of these can influence your germline, and whether these have consequences for subsequent generations. We can learn about human development using this kind of a scheme, because we can make different tissues. And I think very importantly, one of the more exciting things, and certainly that's the thing that we are very excited about in the lab, is can we use this kind of a system to understand aspects of evolution, particularly human evolution, and aspects that make us human, particularly our brain, the complexity of the brain in Arnold. We have a fantastic talk. There are other aspects to this, and we are particularly interested to know whether some of the non-coding DNA that is transmitted by the germline might have been used to induce changes in the developing human brain. So I want to leave you with members of my group. This is my current group. Over the years, many people have worked in my group. I would like to introduce João Alves, who is my first Portuguese postdoc in the lab. He's just been there for a few months. He's doing very well. He's working on germinoids. And I want to thank particularly my funders for a very long period of time, Wellcome Trust, Medical Research Council, and others, and some of the collaborators. Thank you.